Okay, so this is going to be hopefully a relatively brief single lecture on just the basic concepts of expression and differential expression or abundance estimation. You've already looked at a view like this, right? So you've already started to get a sense of what it looks like to see your raw RNA-seq data now aligned to the genome. And here we're looking at a single gene locus, right? And this is for 2 samples. And you can already start to infer some things just from this kind of high-level zoomed out view of this gene, right? So one of them is that there's a lot more coverage up here, right? So this thin track up here is like giving you a sense of coverage. And I think they're set at the same scale. So these bars are higher up here for this sample than down here for this sample. If you were to scroll up and down in this alignment view here, you would see that the reads kind of run out much sooner. There's like a lot more reads going down here, and that's reflected in these coverage estimates up here. And also you can see in this particular sample, there's definitely suggestion of some pretty bad end bias, like you're getting a lot of reads over here in the 3' exons, and it's kind of tailing away almost to nothing at the 5' end. But putting that aside, you might surmise that this particular sample on the bottom is downregulated or like down expressed relative to this sample up here. But of course, we can't do that in a sort of manual way across all the genes. In the genome, we need like a systematic way, a robust statistical approach for doing like what we're kind of intuiting by looking at the data here. And so that's what we're going to talk about. Really, the very first time RNA-seq was done, way back when, people already started to realize like there's a challenge that you can't simply just look at the overall coverage or just count the reads and compare them between genes or conditions or samples because there's at least 2 sort of obvious problems. So one is that genes are different sizes, right? So if you have a big gene and a small gene, and you randomly fragment the big gene and you randomly fragment the small gene, and then you sequence from them, you'll get more fragments from the big gene, right? Because it's bigger. So you can't just like count the reads and say, oh, this gene is more highly expressed, when actually it's just bigger. So you have to deal with that problem somehow. And then you have the other problem that the library sizes could be different, right? Like if we were just looking in IGV and we're like, oh, there's way more reads for this sample, that gene must be upregulated, but we sequence that sample 5 times as much, that's not really true either, right? Like the more fragments, the more alignments might just represent the fact that like the sequencer ran for a lot more reads, that you just got a deeper library for that sample. So you have to account for these 2 obvious things. And FPCAM was like, or RPCAM was the sort of first simple way of dealing with this, which is just to normalize per kilobase of transcript or gene size and per million mapped reads, which is a measure of the library size. And the RPCAM versus FPCAM, it's just a terminology thing. When we started, we were just doing single end reads. But then when we started doing paired end reads, we didn't count the 2 reads twice. We counted it as like one fragment, one paired end sequence fragment. So that's why we started saying FPCAM. And this is just kind of explaining what I just said, right? So why not just count the reads? It's because you have different gene sizes, you have different library depths. So you're going to normalize for the size of the gene or transcript and for the size of the library. And so we can kind of just define a simple formula where C is the number of total mappable fragments for a gene, N is the total mappable fragments in the whole library, L is the number of base pairs in the gene, like the gene or transcript size, and then we could just calculate FPCAM with the formula below, C over N times L times 1,000 times 1 million. There's a whole bunch of different ways to represent this formula. That's just like, there's different ways to do math. You might have heard of TPM. So I would say at this point, TPM is maybe edged out FPCAM as a more popular metric for estimating expression levels, and they're very similar. There's just like a slight difference in the order of operations. So both of them are normalizing per million reads of library. Both of them are normalizing per kilobase of transcript or gene size. They're just doing it in a subtly different way that has an important consequence. And that consequence is that the sum of all the TPMs in every sample where you calculate TPMs is the same. So the way the TPM formula is set up, when you add up all the TPMs for a library, they add up to 1 million. And that is useful because, and it's not true for FBCam. And that's useful in theory because then when you compare across samples, the measures should be like a little bit more comparable. They're sort of like proportional to the same denominator, if you will. In practice, like we're going to show you some correlation plots, they're often like very, very highly correlated. So it's like kind of maybe formally better in some ways to use TPM, but it doesn't necessarily always have a big impact. So we're going to use StringTie to estimate FBCam or TPM values. StringTie is a pretty sophisticated piece of software and algorithm. It does a whole bunch of different things. And the reason is that it's trying to solve the problem essentially of not just estimating gene expression, which by comparison is relatively easy. It's trying to solve the problem of isoform level expression estimation, right? It's trying to figure out not only how expressed is this gene at the locus level, but which specific isoforms are being expressed and at what levels. Like, is all of the expression output from this gene one specific isoform? Is it a mix of these two isoforms? Is it spread across 3 isoforms? Does that change between different conditions? So if you're interested in like transcript level information, which ultimately we all should be, because there are like pretty significant important biological differences between transcripts, right? They can have totally different protein sequences and totally different functions. Some of them are involved in completely different pathways that are regulated under completely different conditions. We should probably all be thinking about transcript level. In reality, like 90% of all biology and bioinformatics is still sort of simplified to the gene level, but we should like constantly be remembering that that is suboptimal. And part of the reason why we make this simplification is because this is a hard problem, and StringTie is like a very sophisticated attempt to solve it, but there are some limitations with short read sequences that you just can't always estimate at the transcript level accurately. And then the other problem is like a lot of the downstream tools just don't have that level of sophistication, right? Like to do like a sophisticated pathway analysis at the transcript level rather than the gene level. There may not be like enough information or tools to do that. But nevertheless, it's like kind of important to think about and consider doing transcript level. And StringTie is this like really sophisticated attempt to do that. This is a high level overview of what it involves. So you take your read alignments, so that's what we already have, right? You can optionally assemble them into super reads. Most people skip this step. It does like have some technical advantages, but it's not widely done. And then you map either your reads or your super reads to the genome, and you start building what's called a splice graph, where you look at the exons, either known or inferred, depending on the different modes that you run the StringTie in, that are connected by the read. So like if a read starts in one exon and ends in another exon, that gives you information about the isoform, right? There's like a connection between exon 1 and exon 3 or exon 1 and exon 2. And you use that information to build this splice graph, and then you calculate what's called a flow network. And you basically look for like paths of heaviest flow through this graph, and that's used to estimate the abundance of the different isoforms. And this is like another depiction of that. So it's sort of like a cartoon example, the way we had with with HiSat, where, you know, in this really simple situation where you have this simple transcript that has exon 1, 3, 5 connected. You can basically ask like which exons are connected by the read. So like this exon connects exon 1, or this read connects exon 1 to exon 3. This read connects exon 3 to exon 5, and so on. And you can basically like count the number of reads that support the different connections, like that connect exon 1 to 3 to 5 versus maybe a read that goes straight from exon 1 to exon 5. And then you can calculate like what is the heaviest path, the most supported path through this graph. And then you can estimate an abundance level and then repeat that process for different paths through the graph until you have an expression estimation for the different possible isoforms. So StringTie, it's sort of like HiStat, maybe even more so. Like if you want to understand it, there's probably a whole course there. You would need to know some basic graph theories. It uses some custom heuristics to do things like calculating the heaviest path. There's more graph theory to calculate the flow network. There's optimization theory to figure out the maximum flow. There's lots of definitions and math in the paper if you want to like dig into how it works. But at a high level, it's trying to like really extract, like squeeze all the juice out of the orange that you get from short read sequence data and sort of do the best job possible of getting isoform level expression estimation. There are alternatives to this approach. So that's like kind of the Lamborghini or Ferrari approach for transcript level estimation, which you can also summarize up to gene level estimation. For differential expression analysis, a lot of people just start with raw read counts. So if you're doing gene level differential expression analysis and downstream things like pathway analysis, a lot of people take this simpler path, which is not wrong, to just use raw counts. And they might use a tool like HTC count, like we're going to use, or feature count, to just assign every read to a gene as a count. So the more times reads are aligned to a gene, the more counts that gene gets, and that's like used as a measure of expression. It's only kind of useful together with some of the like statistically robust downstream packages, of which there are several that we'll talk about. Before we get to that, this is kind of a topic that was raised earlier, this idea of like, when does a read count and count towards what? And so you could think about like these different scenarios where the green boxes represent your read alignment. And you have like this sort of simple situation where it's like just in the middle of an exon of a gene. That's kind of easy, right? Like, sure, let's count that read towards that gene. But what if it's like this? Like it overlaps the exon of a gene, but then it also goes into the intron. You may or may not want to consider that a count. And so there are these different modes of running HC-seq that have different level of strictness. We tend to recommend this intersection strict, which for this example, like would not count this. It would say this read is not consistent with like that gene, because like if it's a RNA, why is it going into the intron, basically? Same kind of story for this read, right? Like, why is this read spanning the intron rather than splicing across the intron? So we might not count that towards the gene in this intersection strict mode that we would recommend. But we would count a read like this, right? Which is like nicely spliced across the exons of this gene. Here's an example of a read that could be assigned to gene A, and you're not worried that gene B is here because there's no overlap, right? So this read is nicely contained with gene A, doesn't overlap into gene B, so we can count that towards gene A in any of these different modes. But for a situation like this, you might be less sure because it like overlaps a little with both genes. But again, using sort of logic, you could say, well, this read is consistent with the expression of this gene. It's not really consistent with the expression of this gene because it would be going into this gene's intron. So by some modes, I would count it, and other modes, I would not. And here's a situation where the read kind of is internal to both gene A and gene B, and it's like ambiguous, right? That read could belong to either, except if you have stranded data and gene A is on one strand and gene B is on the other strand, And then you could take that strand information from the read and say, okay, this must be from gene A, which is on the consistent strand versus gene B. And so HT-seq does allow you to provide the strand information so that this should have like an asterisk next to it in the intersection strict mode. You would actually count it if the strand is correct. Question?
Yes, for the second row though, like if you were to be like that strict, like not count it, aren't you removing like possible novel, like alternative splicing?
So the question is for the second row, if we're like using the intersection strict mode, we're ignoring this green read here, which isn't sort of consistent with our basic understanding of how this gene should be kind of expressed. Aren't we losing information about potentially like novel isoforms and novel expression? The answer is yes. And these like count-based approaches that are about like getting gene level estimation kind of throw away all that complexity. They don't really give you even differentiation between known transcripts, and they tell you nothing about novel transcripts. So if you want to learn about novel isoforms, then you need to use like the more sophisticated approach like StringTie or something similar.
Thank you.
And that's kind of why we're doing both. We're going to go through like both paths. So you're going to get like this transcript level delineation, but also the kind of gene level. And there are advantages and disadvantages to both. And that would be like a major like difference that you just highlighted. Yeah, another question.
When you have the overlap with the two genes, why you can't like assign this read to the more abundant gene, like we do with the peptides?
Like this bottom situation?
Yes.
So if you have a read that kind of is contained within two genes, the question is why can't we assign it to the more abundant gene?
Yes.
You could. It's not the way HT-seq count works. It doesn't have like a rule for that. I don't know how I feel about that. Like maybe it makes sense. But how do you know that I guess.
Because in proteomics, for example, we have peptides that are shared between two proteins.
Yeah.
I don't know, MAXIP1, LFQ, all of these things, they assign these peptides to possibly the more abundant protein.
So we're hearing about how like in mass spec, a common approach might be to just like assign sort of multi-mapped or ambiguous data towards like the more abundant protein. And here we could have the same. I think it's a valid question. Like, let's say you have like this read that supports gene A or B, but you have a lot of other reads like all across the exons of gene A that support gene A as being highly expressed. And you have like no reads, barely any reads supporting gene B and any of the exons or exon-exon junctions. It would be a reasonable theory that like this one read that's overlapping the two is actually from the one gene that is clearly expressed and not from the gene that's not expressed. And by not counting it, you're sort of underestimating the abundance of that gene. Like you're missing out on some reads that should be assigned to it.
It's not something that is done by HT-seq.
Not that I'm aware of. Yeah, that doesn't have like that level of sophistication, but you could. We'd have to like, we'd have to read feature counts and HT-seq software to see if they have like an option about that. It would need to like, it would either have to do something simple where it just makes like a simple assumption, like if there's an ambiguous read, just assign it to the higher, or it could do something more sophisticated, like I was talking about, where it could like try and think about like given all the other alignments to these two genes, which is like more probably the gene. Yeah, something like that. Yeah, it's a good thought. I'm not aware, like off the top of my head, of a tool that does that, but it's a valid thought. Any other questions? Okay, so we have expression estimation. Differential expression is then obviously the next step. We want to tie gene expression back to genotype, phenotype in some way. We want to know what genes and transcripts are being expressed at higher or lower levels in different groups of samples. And we want to know, are these differences meaningful, right? Are they significant? And are we accounting for variance and noise successfully? So we're going to do various comparisons in this course, like the UHR versus HBR. and the tumor versus normal and the wild type versus knockout cells, depending on which of the different exercises and assignments you do. We're going to use ball gown as well as one or two other statistical approaches. We're including ball gown because it was kind of developed and packaged with StringTie and HiStat. The way it works is it uses a parametric F-test comparing nested linear models. So it basically fits two models to each feature with expression as the outcome, and one that includes a covariate of interest, like your case or control, or here the UHR versus HBR, and one that doesn't include that covariate. And then the F statistic and p-value are calculated using the fits of these two models. And a significant p-value means the model with the covariate fits better than the model without the covariate, indicating differential expression. There are a lot of different statistical approaches used to determine like a p-value and significance for differential expression. This is just what ball gown uses. One of the main reasons we still include ball gown, even though honestly that package is not very well supported, is it does have some nice visualizations. So like I'm not aware of like this nice visualization for like DSeq, for example, where you can get the transcript level, the isoform level expression estimation. So this is like one of the advantages of using StringTie, right? That we're going to actually get like transcript level expression estimates, and there's like some nice visuals to kind of see, okay, for this gene locus, there are 5 transcripts with these different structures, and it's like especially this particular isoform that we're mostly measuring, but we're also getting low levels of these two and like almost none or negligible levels of these 4th and 5th isoforms. And if you're really interested in the biology of this gene and these proteins from these different isoforms, that kind of level of information might be incredibly important to you. And this might be a nice way to visualize it. So that's kind of StringTie, cummerbund. We talked about the raw count approaches. We're going to use HT-seq to give us raw counts. We can feed those raw counts into multiple packages. We provide worked examples of both DESeq2 and EdgeR, which are kind of competing statistical models for this. I would say now DESeq2 is definitely the most popular, so we'll probably just go through that example. This is kind of an internal debate. Should I use FPKM or TPM? Should I use raw counts? When should I use them? I'd say the general consensus is you use the FPKM or TPM values when, for one thing, you're wanting to leverage the benefits of like something like the Tuxedo suite, like the StringTie transcript level estimation for getting you isoform level data. They also tend to be a little bit easier to work with for visualization. So like if you're making a heat map, just using the raw counts doesn't work that well because they're not normalized, they're not scaled well. So the FPKM and TPM kind of like just work more easily with visualizations like making a heat map, calculating fold changes. You tend to use the counts when you want to feed them into some of these robust statistical methods for differential expression at the gene level. And in particular, if you have more sophisticated experimental designs, so some of those packages like DESeq, they support, and some other packages that take raw counts, they support more complicated experimental designs like time series or other like multi-condition where you're going beyond just like sort of A versus B comparisons. So that would be another reason why you might go the count-based approach. Generally, multiple approaches are still advisable. This is kind of an outdated Venn diagram. We have updated ones in the course that you'll create that kind of show the level of overlap and non-overlap between these competing differential expression formats or approaches. And they tend to have some overlap, but they also tend to have a fair amount of non-overlap. And, you know, who's to say which one is right? And depending on like your downstream steps, you may want to be more permissive and take like the union of multiple approaches, right? Like if a gene is identified as differentially expressed by both DESeq2 and like ball gown, maybe we include them either way. Or maybe you want to be really strict and say, you know, like I have very limited resources to validate some of these candidates. So I'm just going to go with things I'm most confident in that came out as significant by 2 or 3 different approaches. There are lessons learned from decades ago now about doing expression estimation with microarrays that are still applicable for RNA-seq experiments and for single-cell experiments. You need to consider replicates. You need to think about power analyses and study design. There was this fun paper like way back when that said sequencing technologies does not eliminate biological variability. So this paper came out after like years and years of like really cool RNA-seq studies that had like N of 1 versus N of 1, kind of like no replicates, making like, you know, bold conclusions.
A question from the Slack. How do you perform differential analysis with RSEM data, which is what can be downloaded from sites such as TCGA?
How do you perform differential analysis with RSEM data, which can be downloaded from TCGA? I am familiar with that RSEM is a thing. I've never used it. I think there are like RSEM differential expression packages that are fairly common, and I guess maybe this was part of a pipeline that TCGA used. But I don't know the details of like whether you can extract raw counts. I think you can. And then you could like put the raw counts through these. That's what you would need to do probably. It extracts the raw counts.
Columns, yeah.
Mm-hmm.
Estimated counts and then ppm counts and then ppm counts.
Okay, so it sounds like pretty similar to like the output you will get. Sorry, what was that last thing?
Callisto and Salva.
Yeah, yeah. So similar to Callisto and StringTie, where you get like, you can get both like TPM type measures and raw counts, or estimated raw counts, and you could feed those into like say DESeq or EdgeR. That would be, I guess, the answer to your question. If you wanted to like do differential expression with a count-based approach starting from the RSEM output. Thank you for clarifying that. Any other questions? Okay, so multiple testing correction, you've probably heard of this. It's an old problem. Eventually you're going to get like putatively or apparently significant results kind of due to random chance. And the more tests you do, the more likely you are to get some significant results that are just artifactual by kind of random chance from noise in the data. And so we attempt to correct for that using various multiple testing correction approaches. And RNA-seq kind of just like puts this problem on steroids, right? Because not only do you have 20,000 genes like you would on a microarray, but you have hundreds of thousands of exons and maybe, you know, millions of junctions and introns and microRNAs and link RNAs. Like the tests just keep growing, right? So you can ask so many questions of the RNA-seq data that you're bound to find significant results. And you really need to keep that in mind and think about like, how am I going to like correct for this number of tests so that significant results are actually convincing versus just spurious findings. And there are good packages like bioconductor mult test to help you manage that problem. Downstream of interpretation of expression analysis, this itself could be a whole course. We're going to do many of these things, so we can take expression estimates and differential expression, and we can create visualizations and put them into downstream pipelines to make heat maps and do clustering and potentially do classification, do pathway analysis, and more. And so I think we'll just end there, kind of just reminding you where we are in this workflow. So we have gotten our alignments, and we have run StringTie, or we're about to run StringTie, I guess. We're about to run StringTie. So we're about to start expression estimation and differential expression. And then we're going to feed that. We're going to do 2 paths. So this path, we're going to take our alignments, run StringTie, do ball gown and ball gown visualization. And then we're gonna do this parallel path where we're gonna use our alignments and HTC count to get raw counts and then feed it into EdgeR or DE-seq, probably just DE-seq, and then do DE-seq visualization and other visualizations in R. So any question about where we are and where we're going?